Hello and good morning, everybody. So I would like to illustrate where we are now in terms of Brodmann. Is it still important? So what can we get from this research? And I will give you some examples of how we are continuing this work. Okay. Sorry. Okay. So the human brain, as we know today, is built on multiple levels of organization, and the levels reach from the molecular level or genetic level to the level of single cells with their axons, with their spines, to small circuits, to the so-called mesoscopic level, to the level of whole systems illustrated by changes in receptors and connectivity, for instance, to the whole brain level. And at the end is the brain is not only active as a brain, or cannot be active only as a brain. It needs a body, it needs a periphery, and it needs to realize all the activities, communication. So it's really a complex system about which we talk. And how can we approach such a complex system? Well, one possibility is, of course, to do neuroscience and introduce theories. The other possibility is to analyze data, to simulate perhaps the human brain. But how to get access really to this multi-level organization? And this is not only a question of integrating different techniques, it's really a question that only few of us are dealing with different topics of brain organization. So most of us, or many of us, are dealing with one specific level of organization, and the question is really, can we bring this together? Should we bring this together? And Brodmann's answer would be to analyze the cellular architecture, which is on this so-called mesoscopic level. Well, and he did it by building a cytoarchitectonic map, very similar to other maps that we know from the past. However, in order to build such a map, it is necessary to do tedious anatomical work, including histological processing. And what you see here is a short movie of illustrating how a human brain is being cut into 20-meter thick sections. And you see that the sections are very tricky to be handled. They are a little bit scrambled. Sometimes you introduce folds and so on and so forth. So it's really a very, it's a high quality of work that is needed. And what was done in Brodmann and Vogt's lab, this was on that level done. So he processed, they started to process such sections, and this is, where we also take many of our experience, or most of our experience. And you have seen a photo, or similar to that, by Carl Zilles, and you see illustrated the close collaboration of Brodmann and the Vogt and other researchers. He was the one who became more famous, perhaps, than any of the others, because of his very famous map, which subdivided the cortex into cortical areas, a little bit more than 40, and this seemed to be a very easy map, a very easy map to be applied for nowadays functional imaging, for instance. And Adolf Hopp, Carl Zilles, and myself, we see a little bit us as successors of this work in Düsseldorf, where the institute moved after Vogt's death, and the famous Vogt collection also came to Düsseldorf and is still here, including many of the nice drawings that you have seen in the previous talk. So I'm struggling a little bit with the pointer, but hopefully it works. So when Carl Zilles came to this institute by the end of 80s, he started, and we then continued to analyze the cytoarchitecture based on measurements. And this is a very important point because at Brodmann's time, Vogt's time, there were lots of debates of whether a border should be placed at this position or that position. And in contrast to Brodmann, Vogt, for instance, found more than 150 areas. Von Economo and Koskinas, who also mapped the human brain, they found more than 100 areas. And the question was, is the border really there where they have been described or not? And if you are doing it by pure visual inspection, of course, there are no reliable tools to verify that the border is there, that the cytoarchitecture is different between two different zones. And the only resolution is really to do measurements, which was done here, introduced here by measuring the so-called gray level index, which measures the cell packing density in small measuring fields of 20 by 20 micrometer, and associating it to a certain volume fraction of cell bodies. And this allows us to analyze not only the cytoarchitecture in different brain regions, but also to identify borders based on image analysis, relying on cytoarchitectonic criteria, like here, for instance, when we analyzed the frontal pole and found that differences, for example, in the layer 3, make these areas different from each other. And importantly, this only does not allow to come to a more fine-grained parcellation of the human cerebral cortex, like in this case, where Brodmann subdivided only or distinguished only one area, the frontal pole area 10. And here we have two areas of FP1 and FP2. This is not only a more fine-grained parcellation, it is also a way beyond Brodmann's parcellation, indicating that there are areas, again, with different functions, which are located in this particular position. And the advantage of having image analysis and statistical tools is that we are now in a position to verify the borders independently on the experience of a certain researcher. And we are doing such mapping starting from the mid-90s, I would say, delineating areas like here in 10 human postmortem brains in order to capture intersubject variability of cytoarchitectonic areas in the space. And the areas are being 3D reconstructed and then superimposed in a common reference space of the MNI here, which is a frequently used reference space for many imaging studies. And the probability maps show for each position in the space what is the probability or the variability with which a certain architectonic area is present or not. And such maps help them, of course, to better understand the distribution of areas, and they can be used as anatomical references directly in 3D. And this is an advantage that we have today by using such virtual maps as compared to the 2D schematic drawing. We get a true impression of the 3D structure of an area, of the intersubject variability of the areas, including, for instance, also interhemispheric differences, which are important for many functions that we are trying to analyze. So the maximum probability map is one example how these probability maps can be represented. And until now, we have identified 260 areas. Of these, a little bit more than 100 areas are, 150 approximately, are published, and the half is, so to say, work in progress. And it is really a very labor-intensive and time-intensive procedure also. We are using image analysis. You can see it by the many projects that are going on, and each year costs approximately 1 year. So you can imagine that this work is relying on many shoulders, not only on 1, 2, or 3 shoulders. We had many postdocs and doctoral students. Some of them are present here who contributed to this important work, which we hopefully will continue and finish in a couple of years. My conclusion, however, is that to connect and analyze the more than 86 billion nerve cells with their connections, molecular and genetic properties, a completely new kind of atlas is required, because we have, of course, now access to many more modalities of data which should be included. And such type of atlas is being developed in the context of the Human Brain Project, the largest European initiative for research that the EC has funded. And we are providing in this framework an atlas showing, for instance, cytoarchitectonic probability maps using internet access, and you can select certain cytoarchitectonic areas and then use these areas as a basis to include, for instance, other type of information. So what other types this can be? Well, this can be, for instance, information about the genetic pattern or about the molecular organization, including receptor pattern. So we introduced an ontology which allows you to identify based on brain regions, for instance, and then going to finer grained parcellation, certain cytoarchitectonic areas, and then see and download different types of information. And of course, this is a giant work, that's a huge work, and the atlas is not yet fully populated. We are doing it only time by time, but it shows you a little bit the idea, gives you a flavor into which direction we would like to go when we speak about a multimodal atlas. And to this atlas contributed many of our partners. The International Consortium for Human Brain Mapping was the starting basis of this endeavor. The collaboration with Alan Evan was very important, and now with Jan Bialy, we are technically, methodically implementing this work. We are using also new tools of image analysis to go into the more fine-grained architecture and decoding the inner structure of areas, because we see that although there is a certain homogeneity of cytoarchitectonic areas within each region, so to say, but there are also changes. And some layers, for instance, change earlier than others when we are moving towards the border of two cytoarchitectonic areas, as we did here in area V1 and V2 of the visual cortex. And we are developing new tools in order to parcellate the human brain, for instance, based on self-supervised learning. And it is very important also to verify observer-independent methods by other elements. And there's a second aspect, considering that it's so time-consuming to delineate one area, and if you have 200 or 300 areas, it is a question of time, how fast can we map, and deep learning might be a very powerful tool, and here's some example of the visual cortex to map the areas. And we may also end up with new parcellations of the human brain, as illustrated here by this anatomical clustering, where we have, based on the analysis of very small patches in the human brain, subdivided the cortex into different regions which resemble the lobes of the human cerebral cortex, but this is based just on cytoarchitecture, just cytoarchitecture. I think also, I'm convinced that we can use information about cytoarchitecture, the laminar and the columnar pattern of human brain organization, to inspire new artificial neuronal networks, which are still so much easier and more simple organized than we have in the human brain. And I'm convinced that the heterogeneity that we see in the cerebral cortex is an important feature of the power and strength of human neural networks, which can be perhaps translated to artificial neural networks. Another application that I would identify is to use cytoarchitectonic data much more broadly for modeling and simulation and validation of models. So using quantitative data about neuronal cell density, respecting the different areas of the brain, may help to come up with better models. One of them is seen here for the CA1 region of the hippocampus of one of my colleagues, Michele Migliore, who uses relatively small information at this moment, but will integrate more and more quantitative data, for instance, data coming from receptors from our group. So in my view, we have to team up with computing, because we need large computing power in order to better analyze the human brain, and this is the case because we have so many cells, and if we just analyze one histological section but with a spatial resolution of 1 micrometer, so to identify all these cells, and we imagine that we are introducing 30 virtual planes and have about 7,500 sections, then we end up with 2 petabytes. And this is really a huge amount, and it is only one brain, and of course one brain is not enough for any neuroscientist considering intersubject variability, but still it is an endeavor. It's, in my view, a little bit like a moonshot to 3D reconstruct a whole human brain at this level. It is feasible, however, considering that we have now very sophisticated tools of machine learning, for instance, and what you see here are several histological sections which have physically been cut. And the third and the second dimension, these are virtual sections which have been 3D reconstructed. And what you see now in the zoom in, these are 3D reconstructed cells which physically have been cut and they have been put together again. And this is amazing if you think about that physical cutting introduces distortions up to several millimeters, and we speak here about a precision of a few micrometers. So this is really a hard problem to reconstruct such cells. This is, of course, a problem which creates a lot of challenges regarding computing time, regarding analysis time, I mean, when we have 3D reconstructed the human brain, we can, of course, only, or we will start as neuroscientists to analyze it, which takes really, if we suppose existing methods, several months or several years. But at the end, I would hope that we come up with new models of the human brain like this, based on the so-called Big Brain project, where we can include data coming from ultra-high resolution imaging, like here, cellular reconstruction of the subthalamic nucleus in the brain on a cellular level, and populate the brain more and more with this high-level information to inform other neuroscience projects with such microscopical data. So in my view, cytoarchitecture provides the opportunity to create a conceptual scaffold to identify the principles of brain organization, including cell types, connectivity, microcircuitry, brain function, and at the end, behavior. Cortical parcellation, however, is much more than just putting together a mosaic of area, and decoding the inner structure of this principle is mandatory. Increasing speed and robustness is important. Automatic parcellation of the cytoarchitecture can help us, and to approach the 3 model at cellular or even subcellular resolution, including intersubject variability, includes many questions that have not been solved until more. So we can also at the end learn from cortical architecture to develop new technologies. And so my conclusion is we see so many ways bringing Brodmann's work to the future that I think it will not be boring in the next years. We have a lot to think about how to continue this work, and Brodmann still opens this exciting perspective. Thank you.